So welcome everybody to the afternoon session for Track A, which is focused on digital transformation, data science, and AI. If you were to ask me 20 years ago, these terminologies in pharma, I would say, well, they didn't really exist so much. But nowadays, if you look at the different roles and opportunities, you have many people in the organization with this terminology addressed to their overall title. So it shows the change and transformation which has already taken place for us. For today, we have a lot of interesting speakers as well as great topics for discussion. Steve Steinhubl from physIQ is the first one up on your screen. So Steve, it's a pleasure to virtually meet you, and I wish you a great session and looking forward to hearing your talk.
Thank you, Mo. And thank you everybody for the opportunity. Like everybody, I was there in person. And I wish I could actually attend the other session too, but I'm hoping that when we talk about digital transformation, part of that is also the idea that research can be part of clinical care. I like what Mo said, and 20 years ago, we wouldn't have this. And I started my research career in the mid 1990s during my cardiology fellowship. And let me see if I can make the slide advance here. Which so far I'm not there. I'm not sure if I did that or if it took a minute. All right. So when I was a cardiology fellow, the world's fastest supercomputer at 1 teraflop of speed cost about $67 million in today's cost. Took up an entire room about the size of a tennis court and used the electricity of about 800 homes. If we look at today, just looking at the iPhone 12 as an example, so that's 2 teraflops of speed, 9 watts, and about $800, depending on what you want. And when you think about how clinical research maybe has not changed, there are certainly some tweaks that have occurred in this time 25 years ago between this, but my general theme is I want us to think differently of really what's possible and to kind of recognize how much things have changed. So besides the smartphone and computing power, the connectivity that we have. So this is from Pew Research Center that just shows on the left, home broadband use, which didn't exist at the beginning of the century or just beginning to exist. And now somewhere between where you live, depending on where you live, 72 to 79% of individuals have broadband access. Smartphone use 80 to 90%. This is all in the US. And when you look at just how often people use this connectivity, 93% of US adults use the internet, 85% of US adults use the internet at least daily. And beyond just this connectivity, kind of the new player in the market is these our smartwatches and fitness trackers, which in 2016 was only about 11% of the U.S. population. Now it's about 21% of the U.S. population, and is continuing to grow as the capabilities and potentials grow with these devices. So all of this has led to what is really possible using digital technology to transform research. Now, there are a couple of components of that. I'll go through what I think are the 3 main components of digital technologies that can transform research. But I think a lot of times the first we think of are different wearable sensors. And so this is a figure from an old article now that we wrote just looking at what's possible. And I think that's the best way to think of that, is that I'm going to focus a lot on wrist wearable sensors, but there's almost a limitless potential for sensors and continuous monitoring of individuals that, as long as we think differently and we look at for the value of that we can make markedly improve, I think, our research and care of individuals. These are just some examples of different sensor technologies that none of which existed, I'd say, even 10 years ago in many ways. And so virtually every aspect of your physiology, of some of your interactions with care, like prescriptions and like, can be monitored and tracked. And what I think is, I've, at least in my experience, hasn't been appreciated as much is how that's different, how that can change what we do. Too often we look at these remote sensor technologies as just a way to duplicate what's done in a physician's office or in a research office. So I'll use two examples of this population approach. And so all of us are familiar with, obviously, with temperature, let's say measured oral temperature, and we use a cutoff of fever as meaningful, right? So 100.4 Fahrenheit or 38 degrees Celsius is a fever. Well, you look at this. This is a chart just of 18,000 healthy adults' oral temperatures, and you see there's this broad distribution of that. And so we are essentially ignoring all of this information in an individual and kind of assuming it's unimportant, that this individual variation in their temperature and is unimportant, and then we just do spot checking and we just say fever yes or fever no. But now we have technologies that allow us to continuously track temperature, which in later in my talk, as we talk about cytokine release syndrome and the like, can become really important and in helping identify earlier individual changes and getting away from just this population metric of fever. There we go. Sorry, the slides got mixed up in there. So the same, I'll give the example for heart rate. So this, my colleague, Giorgio Kier, published these data, 92,000 individuals using a wearable, a Fitbit device, looking at resting heart rate. And you can see the distribution of normal resting heart rates. This is not a one-time measure. This is an individual's average resting heart rate over about an 11-month period of time. And you can see that that too, there's this wide variation. With abnormal, there's different definitions, but let's say greater than 100 is tachycardic, less than 50 or some less than 60 would be bradycardic. And so historically, we're used to that kind of spot check. So coming in for an appointment or coming in, for a research checkup, doing an EKG or checking a pulse, and you have that single information. But again, you're ignoring all of this information in between. And now we have multiple devices and different technologies that allow us to track that continuously. And I'm going to show some examples of how that can be important. Sorry, the transition is a little slow there. So when going back to this data set that Giorgio did, so for individuals, every individual has a pretty normal resting heart rate for them. And that can vary, though, between 40 and 100. But for that individual, their resting heart rate rarely varies more than on a median of about 3 beats per minute from a week-to-week basis. But everybody has a different pattern. The top is a postmenopausal woman. The second is a premenopausal woman, and then within the normal cyclic changes on the bottom, you can see there's an outlier of an abnormal increase in resting heart rate, and that could be a measure of some change in the individual's normal physiology. And COVID in particular has given us some examples of that. These are all publications that maybe are not fully appreciated. They're all based on monitoring or tracking individual changes in people's resting heart rate, their resting respiratory rate, their skin temperatures, all through wearable devices. And so this is, I think the COVID pandemic has really started to show us what the potential is for remote monitoring and looking for individual changes in somebody's normal physiology. So not population metrics, not waiting until their heart rate's greater than 100, but seeing if somebody's normal heart rate at rest is, let's say, 55, and now it's 60. So the second arm, which I won't go into as deeply, but is equally important in digital technology, is artificial intelligence and machine learning. And in research, there's virtually every aspect of research that is touched by machine learning and deep learning. Anybody in this field, you can see how it's kind of exploded. This is a graph showing just the increase in the number of studies using either machine or deep learning within the studies, as part of the studies of using that. And all I'll say in terms of that is I think that this is so important. And there was a perspective piece in 2017 that I thought really sums up just in general from research and a care standpoint, that I'll show in the quote, that is acknowledging the profound mismatch between the human mind's abilities and medicine's complexity. So essentially that we have so much data and so much potential for that data that it just in the past, can't be just the human mind that can keep track of all that and take advantage of that. And so really we can use machine learning in virtually every aspect of clinical research, from recruitment to enrichment of patient enrollment, through follow-up, through communications, and then obviously through data analytics. So this is another key part of digital transformation. The last, and I would say the least appreciated in my experience, is the communication part of it. If you think about it, historically, research has been built around these episodes of care, much like clinical care has been, where you come in for a follow-up or you have a phone call with a follow-up with a research assistant. But now if we're incorporating digital technologies and we're collecting data essentially 24/7 through wearable sensors or other sensors, or even just doing intermittent blood pressure checks or other things, we're still connected with our research participant. And that I think it's incumbent upon us to appreciate that and to realize that this isn't just the one-way data transmission that's typically been that we are typically used to dealing with in research, but rather we have to create a two-way data communication if we want to keep our patients engaged and just frankly treating them respectfully. And I think that this failure is best demonstrated by how a lot of really exciting digital technologies and digitally based research trials have shown very poor stickiness. This is a really nice paper that looked at 8 of the original or the earliest research kit studies that often had tremendous enrollment, 50,000 people in a week and the like. And you'll see in these 8 studies that median participation across all of them was only 5.5 days. And it varied between 2 and 26 days. And some of that, some of that usage, so this is shown here in these plots, which are really interesting. So it varies differently between, and you were able to classify people into dedicated users down to sporadic users, and it really shows that it's possible through different addressing, I'd say, different behavioral economic kind of questions and asking how do we return value, how do we keep participants engaged, because we're just not used to doing that. We're used to doing it somewhat through brute force. And with digital connectivity, we're kind of mandated to do it differently. Sorry about the slides. So this is just another example of the same with the device, not only just app-based. This is a study we ran that Dr. Jill Whalen wrote, and we had a very innovative device. We actually had an incredibly engaged population because it was people who had actually invested in this device. And you can see even with that, just giving people information, it was an EKG, their blood pressure, respiratory rate, heart rate, obviously. The stickiness kind of wore off. So people after 17 weeks, that was the median drop off of people just stopped using it anymore. And so we often kind of assume that we give people the devices and that they will use it every day when we want them to, and that's not the case. So this leads to what I think a lot of people have experienced so far is this kind of equation of old research organization plus new technology just leads to costly old research organization, and that goes for academic as well as commercial research organizations. And I think that the reason this is analogous to this productivity paradox from a switch from the steam engine to electricity at the late 1800s, where electricity was such a major step forward, but there was actually no perceptible increase in productivity based from switching from a steam engine to electricity. And that's because the systems of manufacturing were designed exactly the same. They didn't take advantage of electricity to build new systems of manufacturing. And it wasn't until that happened, and it happened in Henry Ford and the Model T and the assembly lines. Interestingly, cigarette manufacturers were also very big at using electricity. And it wasn't until they really changed systems of care, or I'm sorry, systems of manufacturing around what electricity allowed, shown here where you saw this marked increase in productivity. And it's the same, I think, in healthcare and healthcare research, where we have kind of the face-to-face episodes of care as the older model, the steam engine analogy. We've used remote episodes of care. So we've taken exactly what we do in a face-to-face, and we've altered that into virtual visits, which is nice. There's some convenience to that, but it's really not fully taking advantage of what's possible through digital technology. And instead, I think if we really build our research platforms around what's possible, that it's not going to look like somebody's participating in research, because they're going to have their watch on their wrist, or they're going to have their speaker in the bookcase and behind that's going to be tracking them, and that they're going to get messages on their smartphone while their research team is going to be passively monitoring data and then giving feedback to the participants. And that is what I think is needed to really rethinking these research. So, when we think about what's possible, when we think that participants will have to determine their own eligibility, and that's going to be really important. I like this article in a lot that is now several years old, but when we recruit people within healthcare systems, we don't fully appreciate that this quote is, patients are often reluctant to assert their interest in the presence of clinicians whom they see as experts. So often patients feel obligated to say yes to join a study. We don't have that in digital studies. And we have to appeal to our participants and let them choose and identify their own eligibility. As I mentioned, there's two-way communication and the return of information. Average iPhone user unlocks their device 80 times a day. So there's a lot of need to, you don't have to communicate that often, but there's this constant need for communications. And then what's key is to provide value back to participants, but it has to be individual value. So it's not the same for everybody. This is a really nice study by Consuelo Wilkins that looked and then looked at what was most interesting and least interesting to participants based on their educational status. And interestingly, genetic information, how genetic it may affect their getting medical risk, was actually the most negative information to give to individuals who had high school education or less. But in those who are higher college degree or more, that was the most important information to return for them. So we have to realize it's just not one blank return of information. So digital technologies alone can't transform clinical research. But if we take advantage of their novel capabilities, new systems of clinical research can be developed around those that can transform the process to one that's more participant-centric, cost-effective, and real-world relevant. So I want to transition then into specifically work at physIQ that we're gearing towards in immuno-oncology, how wearables and predictive analytics, taking advantage of these individual data and changes for that individual, how it can improve the patient care. Just for those who aren't familiar, CAR T therapy is I'm going to focus my examples on this, is a huge breakthrough in treatments, especially for hematopoietic cancers. There are about 500 CAR-T cell trials going on right now. One of the most common side effects and problems with CAR-T is cytokine release syndrome. In general, cytokine release syndrome, just think of it as pro-inflammatory cytokines that are released. Those trigger more pro-inflammatory cytokines. So it leads to this cascade, which can end up leading to a cytokine storm and very severe illness. Right now, this cytokine release syndrome is graded into 4 grades, but the initial symptoms or the signs is a temperature of greater than 38. So again, a population metric. Now, why CRS is important to identify early, because we can improve therapeutic outcomes, because one is it can be confusing. There are other things going on besides CRS because there's lymphodepletion therapy first, there's a risk of infection. But importantly, as shown on the right, there's not only existing therapies, in particular IL-6 blocking agents, but there are others in the undergoing study and evaluation. So there's a lot of potential to treat CRS once it's recognized. I mentioned, we're using this population cutoff of a fever of greater than 38 degrees as the first measure, and I think there's evidence that we can do much better than that. This is a study that just is looking at inflammation due to LPS injection. On the right is increase in TNF alpha, which you can see at 1 hour is already near its peak. And then tracking temperature change and heart rate changes. And I'm going to focus on the higher doses. But you look at, so 30 minutes after LPS injection, the temperature's increased only 0.1 degree Celsius, 60 minutes, 0.2, 90 minutes, 0.5. By 30 minutes after this LPS injection, the resting heart rate has increased by 10 beats per minute. By 60 minutes has increased by 18 beats per minute, and by 90 minutes by 24 beats per minute. So heart rate is just heart rate alone, but having to know that individual change in heart rate is actually much more responsive to an inflammatory response or a better marker of an early inflammatory response. So you think of what we do within physIQ is we can use multiple parameters. So with a patch, and then with intermittent blood pressure and oxygen saturation, that can lead to multiple different metrics that you're measuring on a minute-to-minute, even second-to-second basis. And in work that we've done, that actually leads to greater than 350 distinct features per hour. And then we use those features in our personalized predictive analytics that we call Accelerate IQ. And with this system, basically, you learn somebody's normal, then you can look at the baseline performance, able to look at deviations from that. And again, this is in as many as these 350 features. And this leads to our FDA-cleared multivariate change index as an indication of an individual's physiology being way out of whack from what their normal physiology is. And this enables a monitoring program that allows for outpatient safe monitoring with continuous monitoring that includes body temperature, heart rate, respiratory rate, activity and sleep, then all the combined metrics with that, along with blood pressure, oxygen saturation. You can look for neurotoxicities through audio and digital testing, as well as symptom surveys. We have data not in the CRS population or in a CAR-T population yet, but in the COVID population to help demonstrate the capabilities of monitoring individuals continuously and leading to early alerting. These are data in phase 1 with 400 individuals showing people wore the patch for a month at a time and had very good data, 96% with that. these are all individuals who were diagnosed with COVID and were sent home, and we're monitoring was looking for the early prediction of the need for rehospitalization. These data on the left show in orange when a COVID decompensation index alert predicted a multivariate modeling based on the patient's individual changes. And if you look at alerting in this one example of a patient that the CDI starts increasing 48 hours before there's that rehospitalization, and if you look at just heart rate or respiratory rate and changes there was no significant changes that would have triggered, especially in a population range, that would have triggered a hospitalization just based on that. And when you look at the phase 2 data, the predictive analytics was able to give a lead time decompensation on a mean and median of almost 2 days before, and with a maximum lead time of 4 days before hospitalization. Similar results in another COVID study with 300 individuals. These was compared to FluPro, but this is just looking at whether they're using individual changes in data to look at positive versus negative for COVID status, and with future work looking at viral load and showing a very strong capabilities of identifying when people were positive and then their improvement shown on the bottom, as opposed to individuals who tested negative throughout the course. And this is preliminary work that is most applicable to CRS and looking at inflammatory response to vaccine. And you can see in orange being able to detect inflammation very early in the hours following COVID vaccine with an mRNA. Now, a lot of the work I just presented is based around patch data. There's a very changing landscape. Just the pictures of the smartwatch actually came from a weekend article in the Washington Post. And there's obviously a lot of advantage to wrist wearables and other wearable devices, but there are also challenges. This is study data in the graph below that shows just to have how frequently you have 20 seconds of continuous good PPG data on a smartwatch, and this is getting the direct raw data, and you can see during the daytime it's only about 5% of the time. In the evening it's about 20% of the time that you're able to get good data. There's also a lot of variability between different devices and the accuracy based on the algorithm. So this is a study from Duke that looked at changes in accuracy in resting heart rate determination and saw the mean absolute errors of up to 14 beats per minute and different through different devices. And but still this is the amount of change and the improvements in the device over time has been remarkable. And so we do a lot of work with the Samsung Galaxy 4 that has remarkable capabilities and other technologies that continuously track and watch changes. So in summary, I think CAR T cell therapies hold exceptional promise as an unprecedented life-saving therapy. However, the cytokine release syndrome is a major barrier to expanding for the greater use of CAR T cell therapy. That the use of multiparametric continuous monitoring coupled with the advanced data analytics, though we believe can provide very early individualized diagnostic and prognostic information that can improve the safety and overall outcomes in patients treated with CAR T and other immunotherapies. I want to thank everybody for the chance to present. Again, I wish I could do it in person. If there are any questions, they'll be sent to me, and I'll be very happy to answer them and discuss and get back to you with that. So thank you for your time.
Thank you, Steve. That was very helpful and extremely interesting in terms of seeing the overall trajectory and how even now with modern applications with immuno-oncology is being used. We did have a couple of questions in the chat, so they'll be sent to you as you mentioned.